Here we'll discuss about the different separation techniques. These techniques will be used to separate the components of a mixture. Let's take, for example, this mixture. So it's composed of different components. How do we separate these different components? Well, I could just look on their colors. So I could pick the blue one from the violet, from the orange one, from the yellow one. So here, I can separate them based on their difference in color. And that's true for other separation techniques. So the basis is on their difference on their properties. Now let's check the other types of separation techniques we can use in the laboratory or in day-to-day activities. This one is a suspension of sand and water. Now how do I separate the components of this mixture? Well, I could do filtration, so I could use filters to separate the sand from the water. Here, we'll use some filters. It can be a strainer or it can be a filter paper. So this one is a filter paper and this one is placed on a funnel. So this is the standard tool that we use in filtering different chemicals in the or mixtures in the laboratory. Now the basis for the separation here is based on the differences on their particle size. So here, the particle size of sand is large, so it cannot pass through the pores or holes in the filter. And here only the particles of water can pass because the particle size of water is small enough to pass this filter. So that's filtration. And by the way here, yung liquid na makapass through the filter, we call that one as the filtrate. So here, the water is the filtrate. Meanwhile, yung magstay dito sa filter, we call that one as your residue. Now let's have another one. This one is a mixture of rocks and sand. How do we separate this? We can do the same thing. We can filter this one, but here the term would be sieving. So again, yung filtration, that is separating solid from liquid. Sieving, that is solid from solid. So again, solid yung rocks, then solid yung sand over here. So it's basically the same thing. So merong sieve. So ang tawag dito is sieve. So the same thing, merong hole dito, then yung components with smaller particles can pass through this one and the others will stay. So it's also used in cooking. So when you filter the powder of a flour, so it will pass through the sieve, then the other larger will stay on top. Let's go back to the first mixture. So this one is again a suspension of sand and water. Aside from filtration, in what other ways can we separate this one? Well, of course, over time, lahat ng sand will settle sa baba. So it will settle sa baba ng container. And then that's because they have different densities, diba? Of course, mas dense yung sand compared sa water. Now, after this, I could just pour this one, the layer ng water, to the other container. So we call this process as decantation. So again, you just pour the top na layer to another container. And in this case, by the way, ang tawag aning liquid na gitransfer to another container, we call this one as the supernatant. Again, this is your supernatant. How about this one? This one is a mixture of oil and water. Now these two liquids are immiscible. Immiscible means they do not mix. So here, what do you think will happen over time? So again, they won't mix. So here, after time, it will form a layer. So yung oil, it will stay on top because here the oil is less denser compared to the water. So again, over time, we will have two layers. They will form layers because again, they do not mix. Yung taas is ang oil, then baba yung water. Then after that, I could just do decantation. But here, I can use a special tool, which is your sep funnel or a separatory funnel. So again, it will form these layers over time. then yung oil will stay on top, then water baba. Then I could just open the valve and I could now release the water. So this, after that, I now have separated the oil from the water. Next, we have blood. How do we separate the components of a blood? We can do a special process called centrifugation. Here we use a machine called centrifuge. And to see how this works, let us watch this video.
You can separate components of a solution such as plasma and red blood cells from a sample of blood. In a lab, tubes of blood are loaded into a centrifuge in pairs. Samples are loaded in opposite positions to help balance the centrifuge. The spinning forces the heavier components, such as red blood cells, to separate from the plasma solution and collect at the bottom of the tube.
Okay, so that's the centrifugation. So here we take advantage of the centrifugal force, and that force it can separate the different components in the blood. So let's say after the centrifugation, you will now have these layers. So let me just clarify. So this one is full na blood, by the way. Again, before we have the centrifugation, it is something like this. And then after sa pag-centrifuge, we can now see the plasma. So here, it forms layers. And again, this is also based on the differences on their densities. So here yung denser na components will be here baba. Then yung less denser will stay on top. Next, this one is a mixture of sulfur and iron filings. Here, iron are attracted to magnets. So here, I could use a magnet to separate the iron filings from the sulfur. This one is magnetic separation. Next, this one is a saline solution. So this is the standard term, by the way, for salt and water solution. Because when I say salt water, this means the water that we can get sa sea or sa ocean, right? And of course, this also includes some rock debris and also some dust, right? Some dust and other microscopic particles. So again, when it's just salt and water, only this components, we call that one as a saline solution. So how do we separate this? We can do evaporation. So we could place this one on an evaporating dish and then we just boil this one. So here, the differences on the boiling point can be used. So of course, ang boiling point ng water, again, the boiling point ng water, it's of course 100 degrees Celsius. And then yung boiling point ng asin or ng salt, it actually boils at 1413 degrees Celsius. So let's say here when we heat this one, so the temperature becomes 100 degrees Celsius. So again, that's the boiling point of water. So at this temperature, mag-evaporate na yung water. Then again, magstay lang yung salt over here because again, it will not boil because it will only boil when it reaches this very high temperature. So this, we have now separated the water from the salt. Another mixture here, we have a solution of ethanol and water. This, by the way, this one is, these liquids are miscible. So these liquids are miscible, meaning they mix together. So how do I separate the components of two miscible liquids? So here I could do a distillation. So here we take advantage of the difference on their boiling point. So the boiling point of ethanol is 78.37. Then for water, it's about 100 degrees Celsius. Now here we do this setup. So we'll also do this one in our laboratory next week. So here we place this solution over here. Then let's say we add a flame here. Then we make sure that it's about 80 degrees Celsius. So on this temperature, which one do you think will boil and will evaporate? So again, it has reached the boiling point of the ethanol. So at this temperature, the ethanol will now evaporate. And again, the water will not evaporate because it will only evaporate by this temperature. So again, this is a mixture. So it's still separated na water, then it's still separated na ethanol. So they are not of the same boiling point. So again here at this temperature, so I need to check the temperatures. We also had uh, add here a thermometer to check the temperature to make sure that the temperature is below the boiling point of the other component. Again, it will evaporate. Then here we have a condenser. So this will cool down. It will cool down the vapor. Then the vapor will turn back to liquid. And then by that, we can now collect the ethanol. Okay, so that's how we do distillation. Again, we take advantage of the difference of their boiling point of the two different liquids.